today we're diving into a hidden world that's literally all around us. I mean, it's in every drop of water, every single handful of soil. It's this enormous, invisible library of chemicals, and for the very first time, we're starting to figure out how to read its books, and more importantly, how they affect life itself.
So let me just ask you this. Have you ever really stopped to wonder what's actually in the water you drink or the soil that's growing your food? I mean, yeah, sure, there's the stuff we can see, but beyond that, there's this incredible chemical complexity, a cocktail of millions of different molecules that, well, up until now has been a total mystery.
Now, scientists have a name for this stuff. They call it dissolved organic matter, or just DOM. But honestly, I think a much better way to picture it is as a giant natural chemical library. It's made from all the broken down bits of plants and microbes, and it contains millions, literally millions, of different molecular books. The problem? We haven't had a clue how to read most of them.
And that, right there, is the huge challenge. Just imagine trying to find one specific, meaningful sentence in a library where every single book has been put through a shredder and all the pieces are just mixed together in a giant pile. That's pretty much what it's like trying to find the few molecules in this complex soup that actually do something to living cells, the bioactive ones. It's a ridiculously difficult puzzle to solve.
So, how in the world did researchers even begin to tackle this? Well, they came up with this really elegant two-part plan. Super simple, right? Step 1. Make a complete list of every single molecular book in that library. And step 2, test that entire library on living cells and just see what happens. See which ones cause a reaction.
In this workflow here, it shows you exactly how they did it. It's brilliant. See, on the left, they start with all these different samples of dissolved organic matter. Then they run them through two processes at the same time. One path is to get a super detailed chemical profile, and the other path is to expose living cells to those same samples. And then the final step is to bring all that data together to connect a specific chemical to a specific effect on the cells.
Okay, so for that first step, figuring out what all the chemicals are, they use this absolute beast of a machine called FTICR-MS. Just think of it as the ultimate ingredient lister. Seriously, this thing is so precise, it can identify thousands of individual molecular formulas from the tiniest little sample, giving them a complete chemical fingerprint of the mixture.
Now, for the second step, they had to figure out what the DOM was actually doing to the cells. And for that, they used a revolutionary technique called cell painting. It's kind of like a biological stress test. You use all these different fluorescent dyes to literally paint different parts of a cell. You know, the nucleus, the mitochondria, its whole skeleton, and then you just take a picture. You get this detailed snapshot of the cell's health and its shape after it's been exposed to the chemical mix.
And this is what makes cell painting such a total game changer. You see, traditional tests, these bioassays, they're super targeted. They ask a simple yes or no question, like, does this specific chemical cause DNA damage? But cell painting, it's non-targeted. It asks a much bigger, more open-ended question. Hey, what happens to the cell's entire structure and health when we add this stuff? It lets you spot effects you weren't even looking for in the first place.
So they ran all the tests. What did they actually find? Well, the first big clue came from this quote right out of the study. It turns out that not all these natural chemicals are created equal, not by a long shot. Some of them, specifically the ones that are more water-repelling or hydrophobic, had a way bigger impact on the cells.
Now this chart shows you the results from that cell painting. Each of those little dots is a population of cells. See that cluster of gray dots? Those are the normal, healthy, untreated cells, our control group. But look over there. See how the blue and yellow dots, the ones treated with specific DOM samples, are pulled way, way away from that healthy group. That visual gap? That's the data, basically screaming that something major is happening to these cells.
So, which samples were causing this huge shift? The answer was crystal clear. The most biologically active ones were something called terrestrial humic acids. This is a type of organic matter that comes mostly from soil and from peat bogs. We're talking about the stuff that is literally under our feet every day.
And the effect they saw wasn't just some vague stress. The cell painting images showed a very specific change. When the cells were exposed to these terrestrial humic acids, the nucleus of the cell, its command center where all the DNA is stored, it visibly shrunk.
So, why is that a big deal? Well, a shrinking nucleus isn't just a minor cosmetic issue. In cell biology, it's a classic textbook sign of major cellular stress. It can be a really early warning that the cell is about to undergo programmed cell death.
something called apoptosis, which can get triggered by things like toxicity or major DNA damage.
Which, of course, brings us to the million-dollar question. The researchers knew that something in those terrestrial humic acids was stressing out the cells, but could they actually use all their data to pinpoint the exact molecular culprits out of that incredibly complex soup?
And the answer was a resounding yes. This is the absolute magic of their approach. By layering the chemical data on top of the cell health data, they could find the exact molecules that were causing the problem. On these charts, which are called Van Crevelen diagrams, you can actually see the molecular suspects clustering together into two really distinct chemical families.
And here they are. The analysis pointed to two main groups of molecules. First, you have lipid-like compounds, so think fatty, waxy substances. And second, something called black carbon-type nitrogen-rich aromatics. Now, these are complex, ring-like structures that can actually be formed when biomass is burned, and they are known to include some pretty nasty environmental contaminants.
Okay, so we followed the breadcrumbs all the way from this messy environmental mixture down to a couple of suspect molecules. Now let's just zoom out for a second and talk about why this whole approach is so incredibly important.
All right, here are the really key takeaways. First, we've basically proven that this dissolved organic matter isn't just a bunch of inert junk. It is a massive, untapped source of molecules that can have very real biological effects. Second, this work shows that cell painting is an unbelievably powerful tool for screening complex stuff without having any idea what you might find. And maybe most importantly, this whole thing creates a new blueprint, a whole new way to hunt for emerging pollutants and to finally start to understand which of the millions of chemicals in our environment are the ones that actually matter for the health of living things.
And that leaves us with this final, really fascinating thought. This study was all about soil and water, but you could use this method anywhere. So what other secrets are hiding out there in our environment, and what else could we learn by painting these unbelievably detailed pictures of life at the cellular level? The library is officially open, and honestly, we're just getting started reading the books.